Hi everyone, my name is Kathryn Atherton, and I'm excited to present our project titled Fun2FITS, a pipeline for estimating fungal community functional gene profiles from ITS-based taxonomic composition. This work is a collaboration between members of the Botniger Lab and the Bioinformatics Challenge Project course at Boston University. Let's start with the motivation behind this work. In microbial ecology, marker gene sequencing, like the ITS rRNA region for fungi, is widely used to characterize communities. ITS tells us what fungal taxa are present, but it doesn't tell us what those fungi can do. On the other hand, metagenomic sequencing can provide that functional insight, but it's expensive and not always feasible at larger scales. In bacterial research, this gap has been addressed with tools like PyCrust2, which predict gene content from 16S sequences using phylogenetic relationships. But fungi are different. The ITS region can't be aligned across the fungal kingdom, so we can't use a traditional phylogenetic tree to impute gene content. However, with the 1,000 Fungal Genomes Project and the expanding Mycocosm database, we now have thousands of fungal reference genomes available. Several groups have started using these references to estimate fungal function from taxonomy, but until now, there hasn't been a standardized pipeline. Our goal was to change that. We developed Fun2FITS, short for Fungi to Function with ITS, a pipeline that predicts the relative abundance of fungal genes in a sample based on ITS amplicon sequencing data. Let me walk you through how the pipeline works. We begin by downloading the list of available fungal genomes from Mycocosm and filtering out any unpublished genomes. The pipeline then matches the fungal taxa in your data to genomes in Mycocosm, either by name or ITS sequence. Once we have those matches, we download the gene annotations from Mycocosm and aggregate the gene counts per genome. These are then averaged to the species and genus level and matched to the taxa in your amplicon data. The next steps use your actual sequencing data. You input an ASV table, and we normalize for ITS copy number per taxon. Then we multiply the gene counts per taxon by the taxon's abundance in your samples. Finally, we normalize each sample by its median single copy gene count, giving you predicted gene counts per sample. To validate Fun2FITS, we ran it on a temperate soil dataset that had both ITS and shotgun metagenomic data from the same samples. Both the predicted and observed gene profiles were normalized using median single copy genes per sample so we could make fair comparisons. Let's look at how we did. First, some good news. Fun2FITS was relatively good at predicting genes associated with ectomycorrhizal fungi, or EMF. These fungi are heavily represented in the Mycocosm database, which likely helps our predictions. As shown in the bar plot, functional categories from EMF genes had higher correlations between predicted and observed counts. Now the less good news. Across the board, we tend to underpredict gene abundances. In the scatter plot of predicted versus observed genes, you can see that our regression line has a slope of just 0.26, and the R-squared is 0.16. This means there's still a lot of variation we aren't capturing. But we also notice an interesting trend. We predict high abundances for many genes that aren't even detected in the fungal metagenome. That raises some questions about what the metagenome is missing or what we're mispredicting. We also wanted to test the utility of Fun2FITS in a real-world ecological analysis. In a case study led by Corinne Viatoras, now in revision at Ecosphere, Fun2FITS was used to predict nutrient cycling genes, specifically nitrogen and phosphorus, in forest soils. The goal was to understand what environmental factors best explain net nitrogen and phosphorus cycling rates, and how important fungal functional genes are in those predictions. Here's what we found. First, the proportion of fungal nitrogen decomposition genes was significantly correlated with net ammonification rates, with a p-value of 0.02. When the analysis was restricted to only ectomycorrhizal fungal genes, the correlation was even stronger, p equals 0.006. This highlights how specific functional groups may drive ecological processes. Second, we found that the relative abundance of fungal phosphorus cycling oxidoreductase genes was the best predictor of net phosphate change in a boosted regression model. As shown in the variable importance plot, this predictor had the largest contribution, over 30%, to model accuracy, even above environmental variables like soil temperature. The corresponding scatter plot shows a strong positive correlation between phosphorus cycling gene abundance and net phosphate change, with a p-value of 0.002. These case study results suggest that even though Fun2FITS is an imperfect tool, it's already capturing meaningful biological signals, especially when focusing on well-represented groups like EMF. In conclusion, Fun2FITS is a first step toward bridging the gap between fungal taxonomic and functional profiling. There's still a lot of room for improvement. Some of the biggest sources of error include missing or mismatched reference genomes in microcosm, especially for less studied fungal groups. That said, our results suggest that the pipeline becomes more accurate for taxa that are well represented in the reference database. We're actively working on 2 improvements. First, we're investigating sources of inaccurate predictions. This work is shown in virtual poster number 2284. Second, we're exploring how to build phylogenetic trees from available genomes to impute gene content even when a genome doesn't exist. See virtual poster number 2192 for that work. Importantly, because Fun2FITS pulls from the most up-to-date version of microcosm, the pipeline's accuracy should naturally improve as the database expands. We're excited to hear your feedback, ideas, or critiques, especially as we aim to improve this tool for the broader fungal research community. Finally, I'd like to thank Chance Knopfsinger, the bioinformatics master's student interns, and our mentors and lab mates for their input and support, and our funding sources, the Boston University Microbiome Initiative, NSF Urban NRT, and the NIH T32 Bioinformatics Program. Thanks for listening, and feel free to scan the QR code or reach out if you'd like to try Fun2FITS on your own data.